something I'm kind of interested in more recently. I've been working in diffusion MRI for quite a while, but this is a different topic. It's about modeling individual variations in task fMRI. And so the talk is about a very simple question that I asked a couple of years ago, which is whether we can take a task-free scan, and I'll define what I mean by task-free scan later on, but a scan where people aren't actually instructed to do any task. And from that single scan, can we predict how they will respond if we were to ask them to do a task? It's a very simple question, and why we want to do that, there's several different interesting things about it and applications, which I will talk about throughout the talk. But that's the simple question: can we take a single scan and predict several different responses to scans? And crucially, I was also interested in finding out whether we can do that at the individual level. So predict variations between people in these kind of task scans. I wasn't sure if the audience was familiar with neuroimaging techniques, so I have this silly slide that tells you about what task MRI is. I'm sorry about this, but very quickly, just in case some of you are outliers, in task MRI, typically we stick someone in the scanner. We ask them to do a controlled task. For instance, in this case, this subject was shown visual stimuli or given auditory stimuli alternating. You can visualize this data in, for instance, this viewer. If you select a point in the brain, you see in red data going up and down with the stimuli, and you can model that and find out brain regions that respond to the tasks. And this is a paradigm that's widely used to, for instance, link brain activity with behavior, and among other things. So that's what I mean by task fMRI. I mean the spatial pattern of activity that you see in individual subjects when they're in kind of an evoked task context. And when you do task MRI, you witness a lot of individual variations in how people and how these maps look like. So what are the sources of such differences? To me, there are three broad categories of sources for differences between people. The first one is kind of boring. People or brain images can have different noise sources. For instance, imaging artifacts that are inconsistent between people, or physiological noise-induced effects in the data. And that introduces variation. A second type of variation is people might be doing different tasks. Even if they're instructed to do the same task, they might not be task compliant, they might not have understood the task, or they might be using different strategies in how to solve a particular task. And that may introduce variations in these brain maps. But the effects I was interested in is whether we can see differences between people just because they have different brains. And by that I mean the brain connections are different, the brain networks are in some way different, and that gives rise to variations in brain maps. So for example, if one person uses more of their frontal lobe in a task, I want to know whether that's because their brains are connected differently or because they think harder about a task or something like that. Okay, so so that's the kind of main goal of what I'm going to show today. But to step back and think about how connections relate to brain function, it's been well known and quite an influential idea, the fact that brain connections to some extent determine or are functional signatures of brain areas. And when you think about it, it seems obvious that connections determine the kind of incoming and outgoing information to each brain area, so you can use them as proxy for fingerprinting areas in the brain. What I'm showing here is just extracted from this paper here, which shows a connectional fingerprint for 2 brain areas in the monkey, area 14 and area 9. And these are the quantitative connectivity from tracer studies to other brain areas. And the point here is that area 14 and 9 have different such fingerprints. And you can measure connections in vivo in humans with MRI, and this has been used extensively to parcellate the brain into different functional regions based on this idea that connections determine brain function. And these are just a few examples of such research. A few years back, this Kanwish's group, in particular Zainab Sayin, published a paper where they pushed this idea a bit further. They reasoned that if brain connections determine the borders between brain regions, then surely if you measure these connections in individual subjects, then we can find these brain regions in individual subjects. And that's what they've done here. They use a task fMRI paradigm where people are shown faces or scenes, and these activate these adjacent clusters of brain regions in the fusiform gyrus. And they used a model that learns relationship between connectivity and task fMRI, and then applied this model to individual subjects to find variations between subjects that kind of mimic the variations you see in the actual task data. And like to give you more detail about how this works, imagine this is your fusiform gyrus here, and it has some connections to areas A, B, and C. And each subject will have such a pattern of connections, but they will look different in different subjects. And what their model did was take your fusiform gyrus with the true brain activity in it, and model the relationship between that and the pattern of connectivity you see to A, to B, or to C. Learn some weights for this model that take you from patterns of connectivity to patterns of brain activity, and apply this to a new subject where you measure the brain connections, you produce these fingerprints, and then use the weights learned in training to predict brain activity. And that's exactly the kind of method I'll be using on different tasks and different connectivity data. So that's the first idea, the idea that connections determine differences between brain areas, and we can take this further to determine differences between people. What I'll be using here is resting state fMRI, which is a technique where you stick people in the scanner and instruct them to not do anything in particular except try not to fall asleep. And it's been shown since '95 that a lot of the co-fluctuations between brain regions in resting state look a little bit like what you'd see in task. For instance, here's a map of a finger tapping experiment. And if you acquire data at rest and look at the signal in the primary motor cortex here, and look at what the signal correlates with, you kind of get something that looks a little bit like the motor cortex, like the motor network seen in the finger tapping experiment, except that it's quite noisy here. But you can do some clever tricks like run an independent component analysis on the resting state data to find kind of cleaner versions of these brain networks and separate them from kind of noise sources. So I thought I would just, instead of just say there's something called independent component analysis, I thought I would have a slide explaining roughly what it does. Again, in case there's outlier people or people who are not familiar with the technique. So ICA in resting state, imagine your brain, your resting state data come in 3D plus time, so a 4D type data set. And if you unwrap space into like one dimension, you can represent your data as a matrix, a space by time matrix, where these are voxels, and each voxel has a time course. And what ICA does is it decomposes this data matrix into the product of two matrices, one made of spatial maps and one made of time courses. And it has a constraint on those spatial maps, the constraint being that these maps are as independent from each other as possible. So non-overlapping, but each map having kind of a characteristic time course. So what you end up with is a bunch of maps that look like this and a bunch of time courses for each of those maps. And it turns out that when you look at the maps, they look like brain networks that you see in task. Some maps are maps of the visual cortex or kind of frontoparietal networks, etc. And you recognize here the default mode network as well. This is kind of a data-driven way of extracting these networks from resting state data, and regions that end up in the same network are regions that you find to be functionally connected if you were to do these correlation analyses. Back in 2009, Steve Smith has published a paper that shows that at the group level, these resting state networks are comparable or look like a lot of the task networks that have been published. And the way they've done that was to take to look at Brain Map database, which is an online database made of quite a lot of fMRI task results, summarized in terms of coordinates of peak activations in the cortex for a bunch of different tasks. And back then it was about 20% of all published task studies, so quite an extensive database. And on the other hand, they had a resting states data set, like kind of standard back then, 6 minutes of rest in about 36 subjects. So both of these data you can represent as a matrix. In the case of this meta-analysis of task MRI data, or the brain map data, the matrix is basically space or voxels by time, but this time is not real time, it's experiments. So you can imagine each experiment will give you a brain map, and you just stack them one after the other. And that gives you a data matrix. And on the other hand, the resting state data also can be represented as a matrix. As I said earlier, you unwrap space into a single dimension, and you get time on the other dimension. And you can run ICA on both to ask whether you can see statistically independent spatial maps. And when you compare these maps, you see quite a lot of them look the same. For example, these are a few examples. In each of these figures, the left-hand side is a resting state map, and the right-hand side is a brain map, so a task map. And that's a visual network. That's also a visual kind of blob. There's the default mode network here, and there's an auditory pair of blobs here, etc. So you can see quite a lot of similarities. And in fact, if you increase the dimensionality of the ICA, so you ask for finer and finer parcellations of the brain with the ICA, you could see that sometimes the two data sets, the two sets of maps split up in similar ways. So for instance, here in this motor network, the kind of the whole network splits into left and right in the same way in both data sets, including the contralateral split in the cerebellum, for instance. Okay, so that told me that there's a lot of similarities between task and rest, and maybe we can use this to find out whether we can translate this into individual subjects. Oh, this is another example, unpublished example, showing if you showing that if you use task to ask where are the borders between brain regions, and if you use resting state data to ask where are these borders between brain regions, the kind of border maps at the group level overlap really, really well. Okay, so back to this question: can we use a single task-free scan to find to mimic task fMRI maps in a bunch of different at an individual level? So I decided to use resting state connectivity to do that. So so before I talk about the method, I'll talk about the data. We use the Human Connectome project data. In case you're not aware of this project, it's another online database which the project started the piloting phase in 2010 and delivered the full or acquired the full 1,200 subjects last year. 900 of those are already released online, so you can download this data. And it's a very rich data set, and the imaging is made of task, rest, and a bunch of structural images as well as diffusion imaging. In total, 4 hours of scanning, so very, very extensive amount of scanning in each subject. And it's completely open database, and there's also tools to process it that are released with the database. And I think the amount of effort that has been put into increasing the data quality in the first stages of the project has had quite an impact on the field. So now everybody is acquiring higher quality data because of the of what they've done for the HCP, which I kind of illustrate with this figure. I think the main thing that HCP has exploited was an acceleration, a slice acceleration technique that is either called multiband or simultaneous multi-slice. But at the end of the day, we can acquire more data per unit time. So for any minute, we can acquire 6 to 8 times more data than we used to. And that allows you to either increase the spatial resolution of your data or increase the amount of data you acquire, or both. He also was aiming to acquire quite a lot of subjects, so to look at variability between people. And 1,200 subjects back then seemed like a big number compared to kind of the typical study. It's comparable to other databases out there, but with the added benefit of data quality being very high. More recently, there's another open data project called Biobank Imaging, which kind of dwarfs the Human Connectome project. So Biobank Imaging aims to acquire 100,000 subjects, and I thought I would advertise it here. The pilot data for the biobank has already been released last month, I think. It's about 4 times the Human Connectome project. In this case, each subject is only scanned for 35 minutes because we've got so many to scan. But the data quality is still very good. Okay, so in the HCP data, there's an hour of task made of several different behavioral domains. So about 2 to 4 minutes per is spent on each of these tasks. And in each domain, you can ask different contrasts between different conditions. So in total, there's 47 contrast maps. So quite a lot of data for testing this idea of whether we can use REST to predict tasks. And this is just showing the spatial overlap between the tasks, so showing that it covers pretty much the whole brain. So I was interested in finding out whether we can. So when you look at the task maps, there's quite a lot of variation, and that's what I was hoping to capture. Okay, so the model itself is very similar to the approach that Zeynep Saygin and Nancy Kanwisher have used with the fusiform face area. We extracted some features from the task-free data, built a simple model that is trained on a number of subjects, and then tested on a kind of left-out subject. So the crucial thing here is that when we predict this map, the model has never seen the actual task data for this particular subject. So that's the point of the leave one out. Most of the features we've used come from REST in connectivity, and I say a few words on how they're extracted, but we also used structural features, which are all listed here. Something we call myelin maps, which are maps that look like heavily myelinated cortex, cortical thickness maps, curvature maps, circle depth, and some diffusion-derived maps, all on the cortex. So very quickly, the feature extraction from fMRI started with a group ICA. So just like in the ICA I showed earlier, where you take your data matrix and you write it as a product of two matrices. In this case, for group ICA, what you do is you concatenate all your subjects and pretend that you have like one subject that has been scanned for a long, long time. And that gives you maps that are representative of the group. The problem with big data is that you can't concatenate too many subjects before you run out of RAM. And this concatenation is necessary if you want to calculate group maps. So instead of concatenating, I use this technique called MIGP, which relies, well, the first thing to say is that before running ICA, we run a PCA on the data. So what this PCA does is basically compresses the data. It takes a matrix that's very big and turns it into a matrix that's quite small, but still contains a lot of the information in the original matrix. For the purpose of this talk, that's what PCA does. Unfortunately, you would need to concatenate, to create that big matrix first before you compress it, and that's the problem. You can't concatenate that many subjects. So what MIGP does, it's an incremental PCA. What it does is that it takes two subjects, runs a PCA, creates a kind of a compressed version of that, and feeds it back as a kind of a new a subject concatenated with another one. So at any time, at any given time, you only have a matrix made of two subjects. And this PCA at the bottom gets closer and closer to what you would get if you were to concatenate all your subjects. So this may look like a technical point, and it is, but when you do big data, you have to care a lot about these kinds of things. What works with 12 subjects no longer works with 1,000 or 100,000. Okay, so the second step after you've got your group ICA maps is to turn them into individual subjects maps, and I use duregression for that. It's called duregression because it's a pair of regressions on the same data set, which is the individual subject data. So you take your group maps as a kind of, in a GLM, regress them against the time series data to get time courses specific for these maps, and then regress the time courses again against the same data to get spatial maps that are individualized version of the group maps. These are just examples of such maps. So this is the group ICA on the left. There are individual versions of these group ICA maps from the duregression. And I've added one more step, which is to use these maps in the middle to create connectivity maps. So it's a little bit like the seed-based connectivity I showed earlier with the motor cortex, except that the seeds are not placed by hand, but they're placed by using this combination of group ICA and duregression. But what you end up with at the end of the day is features that are calculating connectivity, brain-wide connectivity. Okay, so examples of fMRI features on the top, structural features on the bottom for a given subject. Okay, again, just to repeat, these features are fed into a model that is trained on n-1 subjects and then applied to a left-out subject. Now, some results. This is one of the tasks. It's a theory of mind task, and what you see in here is three different subjects that have quite different activation profiles, particularly in the frontal lobe. And I'm zooming in here in the frontal lobe. So the top bit is the actual maps, and the model, the predictions from the model kind of capture these intricate variations between subjects. So I'm going to show more task contrasts here. So that's the one I've showed earlier, the theory of mind contrast, but it also works in the language task, the motor task, and this relational task. What you can do is also like qualitatively visualize this overlap and compare it. I want to compare it to some sort of baseline or kind of null model. So for that, I've chosen what I call here canonical subject, which is like a subject that is bang in the middle, looks like everybody else. I thought that was a better baseline than averaging all the subjects, because when you average all the subjects, suddenly kind of the noise properties of your spatial maps are not the same anymore. What I'm showing here is actual activation in blue, predicted activations in red, and the overlap in yellow. And obviously you can see more overlap, more yellow stuff on the top, where the subject is overlapped with itself, versus overlapping it with the canonical subject. So to quantify that, what you can do is calculate overlap between the prediction and the activation versus self and versus other subjects. So asking the question of whether we do better than picking a random subject. And you can summarize that in a matrix. So this is a correlation matrix where I'm correlating actual maps versus predicted maps for all pairs of subjects. So in the diagonal, you see the correlation between the subject and its own map, and extra diagonally, you see correlations between the subject and some other subject's map. And across a lot of tasks, you see this prominent diagonal, which means that the predictions look closer to the actual maps of the particular subject. So I'm showing this here for 7 different behavioral domains. At the bottom, these histograms are basically the same data. The histogram is the extra diagonal, and the black dots here are the diagonal elements, and you can see that the median of these black dots is always outside of the distribution. If you normalize these matrices, the diagonal pops out even more. So normalizing here just removes the variability between the tasks, but also accounts for the fact that the variability in the actual data is bigger than variability in the predicted maps. I just show that it works across all 47 contrasts, particularly if you normalize the matrices, it kind of pops up. This compares different behavioral domains. So it works very well for more cognitive maps, but it kind of works for all of them. Sorry, what I'm showing here is the x-axis here is this increase. So how much the diagonal is bigger than the extra diagonal. And it's always on the right-hand side. So you always have a diagonal that's bigger than the extra. This is another way of visualizing this matrix, which I find quite instructive or informative. So this is the correlation matrix between actual and predicted for all subjects. And you can represent the data in this matrix in a kind of a low-dimensional representation. So each dot here is a subject, or each pair of dots is a subject, so its actual and its predicted map. And the closer 2 dots are to each other, the more correlated the maps are. So this represents kind of this information visually. You can, if 2 dots are close to each other, those maps are correlated. And I've just linked a subject with its own prediction, just to get a feel for how close you are to your own map. And what this shows is obviously these straight lines are short, meaning that actual and predicted are close to each other, but but also they also short for subjects that are on the periphery. So we're still able to model quite well subjects that don't necessarily look like the kind of group. And that's an information that's kind of not clear when you look at the matrix, but pops out when you look at this low-dimensional embedding. Anyway, so I'm just gonna show some videos that give a qualitative feel for how well these predictions are by just overlapping the prediction and the actual. So I'm going to be showing videos where yellow and blue are positive and negative stats from the contrast maps, and I'm going to be overlapping actual and predicted. So this is a language task with a story versus math contrast. So contrast between a block where people were told a story and asked to interpret it, and one where they were asked to solve a math problem. And I'm scrolling between, I think, 10 subjects in this case. So just to draw your attention to two particular things. For instance, this brain area here, you can see it moving about. So yellow here is the prediction and red is actual. So the prediction kind of follows wherever that red blob is going and kind of stretches and shrinks in a similar way. What you can also see if you concentrate on the frontal lobe is it seems like it's able to capture the amount of activity that's going on. So, so this is what I was mentioning earlier, that it seems like when these people activate more of the frontal lobe, since our predictions are based on resting states where they weren't actually doing this task, it seems like information about that is contained in the resting state connectivity. So it's not that they were thinking harder, it's that their brain are different. So different tasks, yeah, just seems to capture quite a lot of this variability between subjects. Not all of it, it's not perfect, but it does capture quite a lot of variability. And I've chosen this particular contrast. I think it's the emotional task. It has a contrast between faces and shapes, because it seems to be picking up the same region that Zainab Saikin has been modeling with their face contrast. In their case, they were contrasting faces and scenes, but I think the red region here is similar to this one. I'm gonna skip this and this. I'm not quite sure how much time I have left. Okay, so these are the two regions I zoomed on in the video earlier, just to make the point that the model seems to be capturing both variations in what I call location and shape, and variations in topology, so how much of the cortex is activated. And since that's true, I thought I would try to see whether we can predict something that interests many people, which is lateralization of language. And I've calculated an index of lateralization by taking the difference between left and right peak activation. And these are all subjects used here. I've used 100 subjects for this particular set of results. And what you can see is a majority of people are left lateralized for this language task. This is the contrast between story and math. But there's a minority of right lateralized. But what you can also see is that the model is able to capture this lateralization even for people who are right lateralized. And that struck me as interesting because when you do this leave one out analysis, the model is trained on a majority of left-right lateralized people. It doesn't know, I mean, it's not trained separately on the right and the left lateralized, but it's still able to capture this, these guys. So just to delve into more details about the model itself and where the information comes from. These are the beta weights. I'm using kind of a bunch of linear models to make these predictions, and these come with beta weights. And I've ranked them by type of features. So here in this pink box, I've listed all the kind of functional connectivity features on the cortex. The blue bit is the subcortical functional connectivity, and these are all the structural features. So these weights are averaged across all tasks, and it shows that a lot of the like pretty much all features are used to make these predictions. But for any given task, you'll have a subset of such features that are informative. Subcortical signal doesn't seem to be very good in the HCP data. It's very noisy, except for the cerebellum. So a lot of these features aren't doing anything. And I was surprised to see that mean diffusivity at such high betas. But as you'll see in my next slide, if you remove all the structural features and just do the same thing with functional, you get pretty much the same answer. So what I'm showing here is 47 dots, so 47 contrasts, and comparing how well the correlation of a subject with itself, how that measure changes with or without the structural features, and it's pretty much the same. It's slightly lower than the x equals y line, which means that structural features still help a little bit, but they make almost no difference. You can also ask the opposite question: what if I remove all the functional features and only use the structural features to make these predictions? And this diagonal thing disappears completely. And in fact, if you look at the variability between people in the predictions, that variability disappears completely. So this is a correlation matrix between prediction for pairs of subjects. And there's a lot of variation between people in the prediction here, but absolutely no variation if you just use structural. Okay, so like I said, I interpret the fact that we can use a task-free scan to predict variations between people in a bunch of different task domains. I interpret it as as being able to capture this third source of variability, the fact that people have different brains, and that's why the activations are different. I'll finish with a clinical application, since this is the clinical neuroanatomy seminar, and this application is in pre-surgical planning. So the idea is to see whether we could replace pre-surgical mapping using a task with a simple resting state scan, which would be tremendously helpful for both patients and carers. So Natalie Vogt from fMRI provided this data set. It's made of 70 patients and 30 controls. With most of these patients had temporal lobe epilepsy or tumors, but there's also a few others with different diseases. And they all underwent pre-surgical MRI with 5 minutes of resting state and 5 minutes of task. The task was a kind of word generation task where they read a category and they have to think about words belonging to that category. It's a very robust task for activating language networks. So one thing that's interesting about this data set is it's not unusually good like the HCP data. It's actually quite standardly bad. So it's only 5 minutes of resting compared to 1 hour of rest in the HCP. And in fact, it's even worse than this because HCP data uses this fast sampling. So it has a sub-second repetition time, or TR, so 1 volume every 0.7 seconds. Whereas this has a TR of about 2 or 3 seconds. So, so in this 5 minutes of rest, the HCP data has about 4 times the number of volumes that I have here. One interesting thing about this data set also is that there's more variability in the patients than in controls, which is something you would expect, but it's nice to see. And I'm showing it in different ways here with this correlation matrix. So correlating their task maps, you see that there's the controls are kind of more yellow, brightish than the patients. And if you project that into this low-dimensional embedding thing, you see that the blue points, the controls, cluster together more than the patients. So I was interested to know whether training a model on the controls we can get this variability that we observe in the patients. These are just a few examples to show that it kind of it works. It doesn't work as well as the HCP, but it kind of works. You can see these variations between people kind of captured in the model. These are two controls, and these are all patients, including ones with tumors. So we can do the same trick that we did earlier, which is to create this correlation matrix, where on the diagonal you would see correlations between subject and itself, and extra diagonal correlations between subject and others. And if you normalize the matrix, you see that diagonal popping up, like in the HCP tasks. But like I said, I was also interested to know whether this model can extrapolate from a homogeneous population of controls to quite a heterogeneous population of patients. So instead of this leave one out approach that we use with the HCP, where you lump all the controls and patients together and build one model for the left out subject, we've tried to train on controls only and learn a single model that we apply to all the patients. And if you compare both approaches, you see they give very, very similar results. So training on the controls only, which we call transfer learning, or the leave one out, where you lump everybody together. But what's even more interesting to me is that the predictions, so these histograms show how similar pairs of subjects are, and separated into patients and controls, and you can see in the more variation between patients than between controls, even in the predictions. And this model has been trained on controls only. So that's what I mean by extrapolation. Okay, so to summarize, connections seem to strongly predict variations in evoked activity. I've shown in the last example that we can train on healthy brains and generalize to pathological cases. So hopefully in the future we could build models. So if you have a single resting state scan pre-surgically, you don't have to kind of decide, oh, I only have time for this task or that task. You can train several different models in controls and maybe hopefully apply them to patients if that works, if that generalizes to kind of extreme cases where you've got deformations or whatever. And another application that is interesting is to create a tool for functional localization. So using resting state to create task localizers for individual people, which would be quite useful in fMRI research, where people would use a task localizer at the beginning of the experiment to find out where a brain region is, and then do the actual experiment proper. So I was envisaging a software tool called Localize, which doesn't exist yet, but which could potentially provide multiple task localizers provided by a single resting state scan. And I've done a silly exercise of trying to figure out whether such tool could be a money saver. So like instead of spending scan time on a task localizer, saving that money, at least if you have two localizer tasks and you replace one of them with the resting state scan, you can get many localizers. So I plucked these numbers out of thin air, I think about 9,000 publications a year these days, 10 minutes for one scan, for one localizer task. I think about 20% of studies use task localizers. I think more should. And if you multiply all these things together, you get about $6 million a year saved by Localize. So, okay. Probably worth it. Okay, so I just finished by thanking two postdocs that worked on this, Ido Tavor and OEV, and my collaborators at FEMRIB, and in particular thanks to Natalie for the patient data, and to the funding bodies, and to you. Thank you.
Sure, there are many questions.
It looked in your talk like in the motor areas there wasn't as much correlation as other areas. Is that right? And if so, why do you think?
I think for the motor regions, the model was doing well. The predictions were good, but the way I've calculated the overlap using this correlation measure is slightly unfavorable to contrasts where the blobs are small. And if you do the same thing with something like the dice coefficient, dice overlap, then the motor tasks are comparable to the others. Yeah. So I think it's an artifact of the correlation coefficients.
I have a question. How reproducible are these results if you acquire multiple resting states from the same subject?
That's a good question. I haven't investigated that yet. I did look at things like how many subjects do you need for the model to be robust. I haven't looked at whether you get the same predictions if you repeated the scan, but I have the data to do that. So the resting state in the HCP comes in 4 chunks, 2 acquired in one day and 2 acquired in another day. So maybe that kind of split would answer that question. Yeah, good question.
Because I was looking at if I understand correctly how the model works. You're building if I understand correctly how the model, you're building first a sort of model based on the real fMRI task, right?
Yeah.
And then with a leave one out approach, you're using resting state to actually map what are the spatial difference according to for a different subject, right? So the resting state is actually telling us how one brain is different from another most.
Yeah.
That is the very simplified
way
I understand. Okay. Now that's why I was thinking how critical is the resting state data. If you have some changes there, probably you're not going to achieve.
Yeah, so that's an interesting question. If the same could be said about task. If you acquire task data on the same subject on different days, you see some differences. Can you predict that based on the resting state acquired on the same day? So there are questions I don't know the answer to. Yeah.
So when you mentioned that there was 3 components that could be affecting these task maps, one of them was noise and the other one was kind of the connections in the brain. But could there be noise components that are affecting both the resting state and the task data, and so your model is picking up on those?
That's a possibility. I don't see how. So this is why it's important to look at things both qualitatively and quantitatively. If you look at the predicted blobs, a lot of them look reasonable and look like blobs that they don't look like artifacts, basically. So that's one thing. Another thing I could say is because of reviewers, I looked at why some subjects were complete outliers and predictions didn't work, and I found that, as you might expect, data quality affects this quite a lot. So I've calculated 4 or 5 different quality assessment measures on both the rest and the task. And whenever a subject was an outlier in the predictions, it seemed like it was an outlier in one of the QA measures. Yeah, but that doesn't quite relate to what you're saying. What you're saying is, say for instance, there's something about brain morphology, cortical gyrification having some subtle effect on the MRI signal that's causing some artifact. Can it be picked up by both modalities? I think it's an interesting question. I don't know.
I was curious to know, so the analysis was done on the human cognitive data set, the 900 or the 1?
So, because I started this like 2 years ago or something, I used the what was released back then, which was 200.
200,
which, yeah.
Okay. So the question is, do you think by adding another 1,000, you're gonna improve things or not, actually? So the 200, if it works on 200,
Hmm.
Then we can forget about the others.
So the model I'm using here is quite simple, and it seems to stabilize at quite low numbers, well, low-ish.
Okay,
So
that's the
like
second
past 50, I don't think
question. Past 50.
Yeah.
So you could have done. So if it works, any kind of data set that has about 50
Yeah.
subjects is able to show what you were able to show on the 200.
In theory, yeah.
When the prediction of the model doesn't predict the actual response 100%, that part that is not predicted, do you think this is reflecting noise or other factors, or maybe there is something that is genuine to the response of this subject? So what we understand, what we are not predicting, how important is in terms of the actual response, the neural response of this subject?
Yeah, I mean, that's an interesting question. I don't really have an answer. So you're saying, yeah, when we don't make good predictions, can we investigate which category of such differences that belongs to? Like, is that because this particular subject was solving the task differently? Or is it because for some reason the connections don't seem to be predictive in that particular subject? I've looked at something related to that, which is I've looked at the behavioral data during the tasks. Some of those tasks have had good behavioral data, so good variability between people, and it seemed like there was a kind of significant correlation between behavior and predictability. So the better people were at the task, the better the model was at predicting their response. I'm not sure if that tells me that they were necessarily solving the task in the same way. I don't think that's true, but it's a possibility. Yeah.
One thing I was thinking about this model. Is this model kind of universal that you can build all the model from the HCP data or whatever, or a data set with many different tasks, and then you can generate a model and then get a resting state data from somewhere else to do some prediction only? So
In theory, but I haven't tested that in practice.
okay.
In practice, it may not work that simply.
You need somehow some kind of same data quality, same kind of...
Sort of. So, for instance, for the patient data,
Yeah. Okay.
those were short scans, like I said, and I had to modify a few things in the pipeline. So, for instance, in the feature extraction, things like when I was doing multiple regression to extract individual maps, I could no longer do that because I didn't have enough time points. So I replaced that with single regressions instead. And those kinds of subtleties will be data set dependent.
No, I mean, if you can have a kind of an atlas for the model,
Yeah,
and then apply, that would be...
that would be great. Yeah,
Yeah.
but it's still a lot of work to prove that's the case.
I just wanted to ask you the predictions that you made of some of the classic networks that we observe. I think as a sort of quality control measure, it would be nice to see how it spatially correlates with, ICA maps or other well-known networks. Like, for example, I think Neurosync has maps of the default value for several hundreds of studies, and it would be nice to do a sort of spatial correlation with some of those maps to see how well those predictions intersect.
So whether some networks are, whether if you land with some networks, you're better at predicting them, or
Yeah.
is that what you're thinking? Yeah, maybe.
Just as a nice way of
Mm.
visualizing it.
Yeah.
But otherwise,
Thanks.
Have you thought about branching out into different psychiatric disorders, for example, like mood disorders, where you would expect certain changes as well, to see whether individual changes of behavior
Yeah, I mean, not really. Someone contacted me who was interested in, can't remember exactly what the data set was, but it was some sort of drug manipulation, and they were interested to see whether the resting state could capture that. So yeah, I am thinking of expanding in those directions. But those are like more controlled changes. Yeah.
Really impressive accuracy.
Thanks.
I think the best I've seen in a talk like this. Can I sort of clarify related to the question about mood disorders and affective tasks in general? If I understood correctly from your talk, the core functions, the cognitive functions, were better predicted than more affective.
With the caveat that the cost function was correlation. If you replace that with the dice overlap, they're not as impressively better than the model. Yeah. But they're still better, it's true.
And the smaller areas were more difficult to predict. So subcortical areas were very...
I didn't do anything subcortical here, so everything was on the cortex. Yeah. I ignored the subcortex. I thought there was more variability in the cortex. Maybe I'm wrong about that, but it's straightforward to add subcortex to this model.
Yeah. I'm just wondering because typically doing some fMRI research and looking at the individual maps, I personally observe more variations on more affective tasks.
Oh, right, okay.
Tasks. So I'm just wondering
Interesting.
if you'd start taking subcortical areas into account and
yeah, I see.
tasks where
I would love to see your data.
Where there's potentially more individual variation due to maybe arousal levels during affective
I see.
avocation.
What sorts of tasks are these?
Various ones over the years, but I primarily work with schizophrenia, but I also work with meditators, mindfulness practitioners.
Mm.
I'm comparing various affective tasks and self-referential networks and meditators versus.
Well, there is one task here which is kind of peeking into that domain, which is called the emotion task. It's the Hariri task. I don't know how good that is as a model for what you're suggesting. But I should add that even though I'm not making any predictions in the subcortex, the subcortex is still playing a role in features. So I still calculate connectivity maps from the subcortex and use those as features, as cortical features. But yeah, I'll look into that. That's interesting. Yeah.
When you do the leave one out approach, you're sort of saying that this one that I leave out is independent from the rest of the data set that I've trained on.
Yeah.
Does that still hold because you've used all of the subjects previously in one bundle to do this ICA and stuff? Does that introduce any sort of overall correlation statistically between them?
So the ICA, I don't remember exactly. I think the group ICA was done on, I think it was done on a group of 200 subjects. So you're right, it did include that particular subject, but it included the resting state, not the task. And when you do leave one out, it doesn't matter if you include, if you don't include your target, it's fine. But the other thing to add is it's going to have a really minute effect. In fact, I used the same group ICA maps to produce features for the patient data, so it's a completely separate data set. The group ICA is just there to extract features. It has very little effect on the model prediction. In fact, one of the reviewers asked us to replace the group ICA with the random parcellation, and it still works.
Thank you very much.
Okay.
Thanks.